So the next section is the NCBI reference sequences section. So I spoke about this before, so this will give you information about the reference sequences that have been generated for FOXP2. So I'm going to speak a bit about accession numbers a bit later, but essentially we have a couple of different sections here. So we have the genomic section. So this is a section which will give you an accession number linked to the genomic sequence for FOXP2. Okay, and we've got a couple of different links here. So the first one is GenBank. So this, if you click on this, this will open up a new page, and this will be the GenBank entry page for the FOXP2 gene, and we'll be having a look at that now in the next slide. There's also a link for something called FASTA. So this is just a format used for storing sequence data. So you'll see that all sequence data is stored in this particular format. So if you click on this, this will actually link out to a new page giving you the genomic sequence for FOXP2 in FASTA format. Okay. There's also a link to a sequence viewer, which is similar to when we had a look at the transcript and the exons and introns. It will give you a view of the genomic sequence of FOXP2 with a visual outlay or visual graphical output. We then also have a section on the mRNA and protein reference sequences that have been generated for FOXP2. So this is showing you a link to the mRNA sequence. If you click on this, it'll open up a page giving you information about the reference mRNA sequence for FOXP2. This will then open up another page looking at the RefSeq protein sequence for FOXP2. And then we have a number of additional link outs to different databases. Some of these are not housed at NCBI. So for example, there's the UniProt SwissProt database, which will link out giving you more information about the functional annotations for FOXP2. Again, UniProt and Tremble. And we'll be speaking a bit about UniProt and Tremble and these databases in the protein lecture, which is the last module, the last lecture of this module. Okay, so what I'm going to show you now is what information we get back if we click on the GenBank entry page for FOXP2. Okay, so again, it tells us here that this is human FOXP2. It says it's a RefSeq gene, so it's a reference sequence for this particular gene has been generated, and that it occurs on chromosome 7. Okay, what we have here is the accession number for the genomic sequence of FOXP2. Okay, and this is the information that we get back. So it tells you that the gene, so this is the accession number for the genomic sequence. It tells you the length of the gene. Tells you that it's the DNA, it tells you that it's a linear sequence, it tells you that the primary sequence was actually submitted on the 4th of January 2015. Okay, again gives you a definition, it gives you an accession number for the genomic sequence. Keywords here are saying that this is a reference sequence and that there is a reference sequence for this particular gene. It tells you the source, so where is it from, humans. Organism, it then gives you the lineage again, and then what you have is a list of references. So these are references for actual studies that have submitted sequence data from which the reference sequence for FOXP2 has been generated. So in most cases there's a long list of actual references, and each one of these references have done some work in generating sequence data for FOXP2 that has been submitted to GenBank, and that has been used to generate the reference sequence. Okay, so on top here, if we click on the FASTA link, remember I said that that is how you actually will then get the sequence for FOXP2. In this case, we're looking at the actual genomic sequence, so this is the actual genomic sequence on the genome. And again, this is the FASTA format that I was speaking to you about. So in case you don't come across this again in the course, the FASTA standard format for storing sequence data is this format, and essentially it always starts with a greater than sign. This greater than sign is then followed by a description line, which is essentially describing what the sequence is. And this can contain various different types of information, but essentially it always has the name of the sequence and the organism that the sequence comes from. And then it's followed by the sequence itself, which is a set number of characters. It's usually 60 characters across, and then the rest of it is just the sequence for that particular gene of interest. So if we click on FASTA, this is what we end up getting. So this is the way that you would extract the genomic sequence for the FOXP2 gene. Okay. So I said before that I'm going to talk a bit in detail about accession numbers. So each GenBank record, consisting of both a sequence and its annotation, is assigned a unique identifier called an accession number. So accession numbers are important because in each of the databases, a particular gene entry or gene entry page will have an associated accession number. Okay. So as an example, I'll speak about this one, and we've actually come across this before. So for FOXP2, we'll see that the accession number starts here, NG underscore and then a number. So this is a unique accession number associated with the genomic sequence for FOXP2. Okay. So accession numbers are unique, and usually accession numbers are different across different databases. But what most of the databases and resources have done is they've cross-linked accession numbers across different databases. So although the databases may have different accession numbers for the same gene, in most instances these are cross-linked and associated, and that's why you're able to link out from one database to another using an accession number. Okay. So in terms of NCBI, and this is in particular just the NCBI, there are a number of conventions that they use in terms of generating accession numbers. And in NCBI, the prefixes tell us what the associated sequence is. So for example, if you see NC underscore and then a number, or NG underscore and then a number, okay, in the RefSec database, this is usually telling you that the sequence is for a genomic region or assembly. So it's the genomic sequence, or it's from some sort of genomic assembly. So it's telling you the genomic sequence for that particular gene. If you see NM underscore and then a number, this is saying that it is the mRNA sequence. If you see NR underscore and a number, this is usually the RNA sequence. And if you see NP underscore, this is telling you that this is the protein sequence. So it is important to understand these prefixes to make sure that you are accessing, when you're trying to access information or trying to download sequences, that you are downloading the correct sequences. So for example, you might be wanting to access the genomic sequence, so make sure that you are actually downloading the genomic sequence and not the mRNA sequence, or vice versa. Okay. There are a couple of computationally predicted sequences that are stored in NCBI, and we're going to be talking about this a bit later in the course as well. And these usually start with X, so XM, XR, XP. Okay, and essentially these are sequences that are computationally derived and are not generally derived from real experimental evidence. So it's just something for you to be aware of when you are accessing information from NCBI. So in the next session, we're going to have a look at basically how we would access similar information from the EBI website. So we'll be using FOXP2, and we'll see how we can access that information from the EBI website and what it looks like at the EBI website.